Everyone, myself Mukesh Verma from India, going to present on title Automated Plant Species Identification using 3D LiDAR Point Clouds. It is a case study using 3D point clouds of the cabbage and maize plants collected from real agricultural fields. Now coming to the introduction part, plant phenotyping is the measurement of the components of the plants and it is essential for the plant species identification. Automated plant species identification using LiDAR technology is an promising approach for precise forest management and precision agriculture. LiDAR data enables the extraction of 3D structural information, individual tree point clouds, and phenotypic traits like leaf area and stem position, contributing to the high throughput phenotyping and precision agricultural applications. By combining diverse approaches, automated plant species identification using LiDAR proves to be a valuable tool for enhancing forest inventory, species discrimination, and plant phenotyping. Two survey-grade instruments, namely terrestrial laser scanner and total stations, are used for high-density precise data collection of the cabbage and maize plants. We have chosen two study areas to conduct our research. Study area 1 belongs to the cabbage plants planted on a sandy loam soil that is covered an area of 450 meters square. The data was collected at the maturity stage of the crop. Another study area was near Sam Higginbottom University of Agriculture, Technology and Sciences, Prayagraj, India. This study area was an agricultural field having maize crop, data collected at the tasseling stage of the maize crop. This is the experimental setup for the data collection. It is shown here. The placement of scanner, total station, checkerboards and spheres can be seen in the diagram. Checkerboards and spheres are useful for the registration of 3D scans obtained from the terrestrial laser scanner. Here I will discuss the overall methodology of this study. After the data collection from the cabbage and maize crop fields, the raw data point cloud data was merged and transformed to a common coordinate system. After merging the 3D scans, the ground points are filtered using cloud compare software. Further, K-means algorithm was implemented to segment each plant from the non-ground points. Then from the individual plants, point cloud, plant volume, projected leaf area, plant heights are estimated for the plant species identification from a common field. The height of each plant was determined by averaging distances between 10 points located at both the top and bottom extremities of the point cloud. Now I will discuss the quick hull algorithm a bit. The quick hull algorithm offers an efficient method for computing the convex hull of a set of points in a plane and a volume. Initially, the algorithm identifies the leftmost and rightmost points as the initial endpoints. These endpoints are then used to form a triangle encompassing all other points in the set. Here we can see the generated 2D convex hull of the individual cabbage plant. Here we can see the 3D triangulation obtained after the implementation of 3D convex hull algorithm and 3D triangulation for volume estimation. The plotted line graph in the figure 13 is depicting cabbage plant height, projected leaf area and volume utilizing 3D point cloud data. We have calculated these parameters to offer insight into the plant development. These findings emphasize the complexity of the cabbage plant growth and make evident the volume of a 3D point cloud analysis in the understanding plant development dynamics. A healthy plant should have optimized values of height, area, and volumes. By comparing the respective height, area, and volume parameters, the plant species can be identified automatically. The variation in the plant height, volume, area can be easily differentiated among the cabbage and maize crops to differentiate between the cabbage and maize crops. Thank you, everyone.